So this morning I'm going to take you on a very beautiful journey to Australia, a country very obsessed with border security and food security. So my talk is basically how can we use genomics and bioinformatics to improve food security and biosecurity in general. And I'm giving you the example of an invasive species, the case of the whitefly Bemisia tabaci. So a very brief background. This is a very invasive species, globally distributed all over the world. And it harbors a wide range of endosymbionts, a lot of viruses. It's very, very serious pest in sub-Saharan Africa. It caused famines and decreases the yield of cassava crop by sometimes 70% in some areas. This is why a lot of attention, especially from philanthropic agencies like Bill Gates Foundation, there is a lot of attention on this problem. So basically, what is the problem? This species, to use general terms, it is a complex of species, which meaning they look morphologically identical, they look exactly the same, but when you look at the genetic material, they are completely different. And this is making the identification at the borders and at the quarantine point of entry very difficult. Therefore, the species is very invasive and we can't manage unexpected outbreaks. So the project was like, okay, 2 decades ago, everybody was trying to identify and do the taxonomy of the species using mitochondrial DNA, which means one single gene. Recently, with the advent of next generation sequencing, we decided to go bigger. So we were like, okay, we're gonna do the whole genome on a genome-wide scale. So we decided to study the genome of the species and try to use these techniques to identify the complex and say how many species in the complex, and therefore improve the biosecurity measures at the quarantine point of entry. And this obviously will help us study the demographic history of the invasive species and try to trace the pathway of invasion all over the world. So we did an extensive sampling all over the world. We traveled, we had collaborators sending us a lot of samples, and we used what we call in simple terms a reduced representation of the genome. So basically we take the genome, we cut it in pieces, and then we have like a view of what's going on instead of taking one gene. Basically this was really hot technique maybe 5 years ago. Right now we're sequencing the whole genomes, but this was really good because we're sequencing a lot of samples, so it's very cheap and practical and very informative as well. So this is basically how we're getting the information across the genome. And of course, any geneticist, the first thing you throw there is a PCA, right? So the PCA shows a very promising result. We identified 3 species, which means this will help us identify them for biosecurity purposes, for border security. So if we receive any samples, we will be able to know which species is which. And we try to understand from a genetic perspective the co-ancestry, the similarities, the genetic relationship, the phylogeny. This will help us also improve the measures for control. We even traced using sophisticated bioinformatic tools when these species split. This is like hardcore evolutionary biology. Like in million years ago, they were the same species and then they split, and we could trace that. I'm very excited about this. So, and we have a statistical backup for the species delimitation, which will strengthen our finding of these findings. So this is also very exciting. So in addition to what we did in the taxonomy part, we used the genetic information on a whole genome scale to understand how these species are moving across the world. For example, if we have samples from France, from Greece, from the United States, using genomic information, SNPs, we can establish the relationships, the migration routes. We can say this sample is coming, invaded France, it's coming from United States or something. So it's a very powerful tool to understand the movement of the species on a global scale, basically. And this is what I was just saying. So the genetic information allowed us to map the routes and the migration of the species across the samples we collected for the study. Very short. So we have exciting findings. This will help us a lot in the decision making, and for this species, particularly at the border security in Australia. They are obsessed with that. So if they find one pest in one fruit, they panic. It's all over the news. And this will help decision making. So if we have the tools, if we have a genome data, we can like establish a SNP ship, like a tool. And at the quarantine point of entry, they are able to identify which species and from where it's coming. And this is powerful knowledge to deal with the problem. I presented this because this is what we have done last year, and just to make a point that genetics and genomic tools are the same. So this is on the edge of public nutrition, agriculture, a little bit of public health, but we can apply the same technique for like epidemiology and for mosquitoes, vector-borne disease. It's very relevant to that, and it will help us map diseases like dengue fever, a project I'm trying to launch with Ali, of course, in the Middle East, MENA region. So just the purpose is we could use all these techniques to really apply them to various wide range of questions, scientific questions. Finally, I would like to acknowledge, of course, the Fulbright program for the strong support. They sponsored my trip, and also I'm very grateful because they co-sponsored my seminar at the UC Berkeley last year. Of course, the German EMBO Fellowship for Molecular Biology Organization, and the Women in Science program, UNESCO L'Oréal. I am also a strong advocate for women in science. We need more women in the room. Dr. Ameni, you know the struggle, you're professor.
I'm happy with all that.
Yeah. So I would love to collaborate in that advocacy area, and if I'm happy to start any collaboration, especially epidemiology right now, dengue fever or malaria or anything similar. And please visit me in Australia.
Thank you.
Thank you.
Samia.